So, there we go.
You're not as loud as that.
Sorry, Webb. But I bring this up, I brought this up at the original HMP meeting years ago, and I continue to bring it up. And one of the reasons you get this, I think, you're getting this extreme host effect with the microbiome, is that it's, these aren't soils. So, we have an immune system, and it's fighting back, right? And I think it's, I have to say that when I see a lot of these studies, and I bring this up, people sort of say, well, yeah, it's a microbial ecology, but I'm just wondering, can we start to get to, and I'm doing this a little bit in my work, but can we start to get to how the host is having some kind of top-down control over the microbiome that might keep it, when it's continually being colonized from outside sources, even from the mouth to the gut or whatever, that it keeps it in exactly the state it's in, both chemically but also through the immune system, cytokines, and everything. And how are you going to get to address that?
Yeah, so that's a fascinating question. One way we're addressing it is by directly collaborating with various people who are measuring immunological parameters ranging from cytokines to T-cell sequencing. The other thing I'll mention, though, is that in studies of the mammalian microbiome, including studies on isogenic lines of mice and studies on monozygotic and dizygotic twins, what we find is that the host genotype has a very small effect on the gut microbiome, at least, compared to diet. So, for example, with Jake Lucis and cell metabolism in 2013, we studied about 50 strains of mice in high-fat and low-fat dietary conditions. And what we found was that quantitatively, the diet effect was a lot greater than the strain effect. And there were also substantial strain by diet interactions. So you couldn't predict where in the point cloud in the high-fat diet a particular mouse was going to be based on its location in the low-fat diet. We've also seen that in humans. So if you look at the Goodrich et al. Cell paper in 2014, when we look at hundreds of twin pairs, we can start to quantify a human genetic contribution. And that contribution is not unimportant. Like Christian Sinella, for example, has a strong correlation with low BMI in that population at least. And if you transplant it into mice that are colonized with microbes from an obese individual, the mice will lose weight if they're co-inoculated with Christian Sinella. But you're talking about a couple of % of variation in the microbial community versus a very strong driver that we see in long-term diet. So I agree with you that the immune system has to be important, but at least in the gut, what we're seeing is that the diet is an incredibly powerful driver there, although obviously many factors are involved in all these.
Rob, it's clear to me that the building microbiome has effect on the human, I'm sorry, rather other way around. It's clear to me that the human microbiome has an effect on the building microbiome. And certainly buildings or microbes in buildings can affect human microbiome through infectious disease. But what evidence, or could you comment on any evidence to sort of show that the building microbiome has some effect on human skin, oral, gut microbiome in ways that are not infectious?
So again, that's a great question. As you know, because the vast majority of this research has been funded by the Sloan Foundation, which is explicitly not allowed to fund health-related research, we just don't know the answer to that question in general. And most of the studies that have been done so far have either been association studies, looking at correlations between, say, the diversity of the microbiology in the home environment and then later incidents of asthma and allergies. Or you also see effects where people who are living in the same environment, and you saw some of this in the data Shelley presented from her collaboration with NOAA on the DAWN project, and we also saw this earlier in the 2013 eLife paper and then in the student microbiome paper, also with NOAA. You see that people who are sharing space tend to converge on their microbiomes, but we don't know if that's a direct effect of the building or whether it's whether it's due to contact between the individuals. And you could say the same about Jess Green's roller derby paper, for example. What I think is going to be really conclusive is animal model experiments where we can manipulate a lot of those variables and see readouts in the short time frame. Many of those have been proposed to Sloan but not funded. I think a really important question is whether other government agencies or other foundations will pick up that line of research, because I think that's what we critically need to be able to directly demonstrate that there's an effect, rather than relying on indirect longitudinal studies. and indirect case control studies where we just can't get at causality because we can't manipulate the variables directly. If anyone knows of examples that I don't know of, I'd love to hear from the audience, but I know this is something that constantly gets brought up in these discussions, and it's the kind of thing that it's very difficult to do without large-scale funding, given the number of samples that you have to collect and the scale of the intervention that you need to do to prove causality in humans.
You went pretty fast. I was fascinated to see the diagram of the person with different fatty acids and how the microbes on the different part of the body affected that, because in so many of our studies, we talk about the different microbes that are there, but we don't talk about what they're doing. And if there are health effects, it's almost certainly through some of these metabolic products. And so I'd just be interested to hear you philosophize about that a bit.
Absolutely. I couldn't agree more with you. That was, in fact, a large part of the reason that I moved at the end of 2014 to work in a medical school across a very small field from Peter Dorrestein, who you'll hear from shortly, who does amazing work on the metabolomic side, and to collaborate directly with immunologists who are able to do the kinds of immunological readouts that it's very difficult to do in a non-medical setting. So absolutely, we need to move beyond inventories and beyond what Leslie Landwander-Boulder is very fond of calling elevator science, where you see things go up and go down, but where do you get off, really? And so being able to get to tractable model systems, including in vitro systems, organoid systems, and animal models, I think is going to be essential. The other big risk that I see is that with the association studies correlating microbes with environment, it's very easy for people reviewing the literature, especially when it has not been possible to fund studies that do a direct correlation to health. It's very easy for people to draw the mistaken conclusion that because no direct causal links have been found that are linked to health, that those links don't exist. Right, and I think especially more effort in that direction from funding sources that are able to fund that line of research. Now that we've learned so much about what's in the built environment, how it changes, what are the factors that are driving it, and how intimate a connection it has to our own human microbiology is really important. There's some really interesting indications. So I worked on a paper with Chris Lowry at Boulder that came out in PNAS earlier this year, where we were looking at Mycobacterium vaccae, which is a soil bacteria. And if you inoculate mice with M. vaccae and then expose them to a social stress situation, they're much more resilient than if they're not exposed to that soil bacterium. And if you combine that work with things like the work on various amphibians, including frogs and salamanders, and the work on the Komodo dragons and other reptiles, suggesting that many different diverse species require a constant input of microbes from the outdoors in order to maintain health in different respects. When you combine that with the diversity data on asthma and allergies in humans, it seems very likely that we'll be able to come up with chemical or immunological mechanisms if there's enough research in that direction. But you have a problem both from the funding side and from the side of assembling the teams of researchers that you need to do that kind of interdisciplinary work.
How do you feel about models lower than vertebrates?
I feel great about those models. Like, I think there's a lot that we can learn from flies or worms in this context, and it's certainly what's been very interesting is seeing researchers in those communities with the incredibly powerful genetic tools that they can bring to bear just start to get interested in microbiome research over the last 2 or 3 years. So I think we'll see a tremendous amount of exciting work from that community.
Okay, so I think Hal's going to be our last question.
Rob, I've asked you this question before, but nobody else except me heard the answer. Could you comment on the importance of the health importance of the human skin microbiome?
Oh, sure, absolutely. So the health importance of the human skin microbiome, and I'm going to rely heavily on Rich Gallo, who's the chair of dermatology at UCSD and doing a lot of work in this area at the moment, as well as Julie Segre and Heidi Kong, who are doing a lot of excellent work on this at NHGRI. In diseases like atopic dermatitis, there's a lot of evidence of the specific role of staphylococcus infections and other that drive community-wide shifts in the skin, changing the balance between health and disease in humans. More recently, there's some very exciting work showing that the skin microbiome, and especially its mediation of inflammation in the skin, mediates all kinds of different diseases, ranging from cancer to the amount of sunburn that you get with a given amount of solar exposure. And there's also been some very elegant mouse work looking at how the microbiome, not just in the skin but also in the gut, helps you resist skin infections when those are experimentally applied. The other thing that I mentioned briefly just before was the work in amphibians, looking at the loss of beneficial skin microbes over time when kept in captivity in a clean environment. And then the amazing part of that, and this was published just recently, is that you can isolate probiotics from them while they're healthy, and then re-inoculate bacteria that would otherwise get, re-inoculate amphibians that would otherwise get chytrid fungal infection with those healthy bacteria that are ultimately environmentally derived. So there's a lot of direct evidence in a range of different species that the skin bacteria, and especially inputs from those skin bacteria, are important for health. Thanks again.
Oh, hi, Scott. I'll just briefly introduce Scott Jackson here. I guess thank you, Scott, for joining us remotely. We've been, the part of the issue that the committee has been dealing with is questions of reproducibility and how this plays into making microbiome measurements, which seems to be a very important topic. And I was sort of very appreciative to sort of follow from a distance a bit the work that Scott Jackson is leading at NIST to look at some of these questions of how to develop benchmarks to measure accurate measurements on microbial communities. And so I'm hoping, I'm expecting he'll be talking about the work that he's been doing in that area. Take it away, Scott.